how can we classify viruses based on nucleic acids? So remember, nucleic acids are at the core of the virus, and we can we can classify viruses based on nucleic acids into viruses with double-stranded DNA genome, or viruses with single-stranded DNA genomes. Then we have some viruses with double-stranded RNA genome, and the rest are for single-stranded RNA genome. Okay. So why do we have three classes of single-stranded RNA genomes? Because one class of them, they have what we call a positive sense. So these are viruses with single-stranded RNA molecule. One way to think of is like a messenger RNA. So these are viruses with a genome that can be very easily and instantaneously translated by the host cell ribosomes into the proteins. The second type is the negative strand, or single-stranded viruses with a minus sense polarity. So they have an opposite polarity to the messenger RNA. They cannot be translated directly into proteins. They need an additional step in order to be translated, and that's the second category. Third category are single-stranded RNA viruses that have the reverse transcriptase enzyme. So reverse transcriptase enzyme will reverse transcribe this RNA genome into DNA. Remember, transcription is a process that used to convert the information encoded in the DNA into an RNA molecule. Then if you go in the opposite way, that's what the reverse transcriptase will do. It will help this RNA molecule to be reversely transcribed back into DNA. So that's broadly the 6 different types of viruses. And if you want to get a visual representation of that, so think about double-stranded DNA like that, two strands of DNA forming the double helix. Then we have single-stranded DNA viruses. Then we have double-stranded RNA viruses. Then we have these 3 types of single-stranded RNA viruses. One of them is with plus sense, or positive polarity, which can be translated directly into protein. The other one is negative sense, so this is an opposite polarity. It needs further steps. Then the last one with the reverse transcriptase enzyme. Okay. So again, one way to think about this group of viruses, maybe you can think of an effervescent tablet and a glass of water. So the effervescent tablet in this case will be the plus sense single-stranded RNA virus genome. So once a virus with this plus sense single-stranded RNA is placed in this cup of water, so which will represent our host cell, what will happen? It will dissolve instantly. That's an analogy of the plus sense RNA genome of the virus once it is inside the host cell. Remember, the host cell is like 70% of it is a water content. So this plus sense single-stranded RNA will be quickly translated with the host ribosomes into the different proteins of the virus. So this is a very quick process, similar to the dissolution of an effervescent tablet once it is placed in water.